Hi, thanks so much for the opportunity to be here today. I am Jitka Virag, and I'm leading Vital Effects to develop solutions that will transform the future of cardiovascular disease care. With Robert, my partner and expert in protein engineering, we've assembled a team that includes experienced personnel in clinical cardiology, veterinary medicine, FDA regulatory guidelines, as well as the executive and entrepreneurial processes that are involved in supporting this endeavor. The problem that we are working to address is that every 40 seconds, someone has a heart attack. Most of you are aware of the impact that cardiovascular disease has, and it's only growing as the population increases and ages. The ischemic heart disease market comprises the largest submarket, and here's why. You were born with all the cardiomyocytes that you will ever have. They do not regenerate, and they're interconnected, so they function as a unit called the syncytium. When part of that unit is damaged, as it is when it's deprived of oxygen during a heart attack, the whole organ becomes dysfunctional. We all have risk factors for heart disease due to heredity and lifestyle choices. If the heart is irreversibly damaged by lack of oxygen for too long, more than 6 hours, the cardiomyocytes will die. When cardiomyocytes die, that area heals by becoming a non-contractile scar. The rest of the heart now has to work harder to compensate for this. And as tissue remodeling continues, more damage can occur, that scar thins, function worsens, so heart failure progressively declines, as shown by the downward curve and the oscillations. After over 50 years and billions of dollars spent on research to address this problem, we still largely rely on early reperfusion or revascularization as our primary means to thwart early damage and disease progression. If this doesn't happen in time, though, or it's unsuccessful, heart failure will progress. We have many drugs that can slow the progression by alleviating the work or the load on the heart, which contributes also to that oscillation, but we still have no cure. Until now, I'm going to show you today how Vital Effects is working to address each part of this problem. Vital Effects solution is VX1. Cardiomyocytes die after prolonged ischemia, 6 hours. When I started investigating EphA1 15 years ago, all I knew was that EphA1 was proangiogenic, and it was the only ligand that could actually bind to all of the receptors. And the receptors are expressed in these unique combinatorial patterns in different cell types, making it versatile for finely tuned druggable targeting. What I didn't know at the time is what became our edge. Vital Effects is based on the endogenously expressed EphA1 protein that's present in healthy cardiomyocytes. We didn't know this when we injected a recombinant version into an injured mouse heart. And what we saw is that it dramatically reduced injury without changing vessel density. Now we are working on to formulate the VX1 for intravenous delivery, determine the time and the dose boundaries relative to the injury onset for an optimal treatment paradigm. And we are also hoping to explore the possibility that it could have prophylactic properties. It could be beneficial to high-risk patients. It has shown robust results in 3 well-established and routinely used mouse models. We've observed complete recovery of cardiac function in acute ischemia with reperfusion. That's that first part at the top of the curve. 50% less damage in chronically non-reperfused heart injury, like the cabbage part of the curve, and improved outcomes following long-term non-reperfusion, possibly by slowing or lessening the slope of that progression to heart failure. This is one dose at one time, a sustained benefit. The efficacy has also been validated by experiments that have been done in 3 other labs using very different approaches. Mitochondrial function in permeabilized fibers that were taken from an acutely ischemia reperfused heart that had been treated 24 hours prior retained function equivalent to those of uninjured controls. After 24 hours of complete anoxia in a dose response study, a maximum of 84% of human inducible pluripotent stem cell derived cardiomyocytes treated with EphA1 will survive. Normally none would survive that. Lastly, it markedly improves ATP production in cells from congenital heart patients, suggesting that it has the potential to be applied to other indications. One of the submarket indications that I mentioned earlier is coronary artery bypass grafting surgery. This surgery requires actually an additional surgery to transpose a vessel from another part of the body that will be used to bypass the one that's blocked in the heart. Given the poor outcomes of this discrete segment of the ischemic heart disease market and that another surgery is required to do it, we are modeling this to assess the potential of FRNA1 in the absence of grafting, which basically equates to a non-reperfused injury. We were awarded a translational research grant by the North Carolina Biotech Center to determine the efficacy of FRNA1 in a preclinical model of coronary artery bypass grafting simulation in rabbits. So far, our results are compelling. The last rabbit of the first goal will be completed tomorrow. We will process the samples and present our results to the NCBC so that they can determine if we're a go for the next part of the study. These results will also be used for resubmission of the phase 1 STTR that I developed with First Flight Venture Center and Eva Garland Consulting in the liftoff program that was awarded to me last year when I participated in their FAST cohort program. The keys to successful attenuation of injury are speed of delivery, potency, and specificity. The faster we can get it into the circulation, the quicker it will get to the heart cells that need it. This will optimize rescue of heart tissue and improve functional outcomes. We believe that VX1 can meet these criteria and will be very useful and possibly even life-saving in a variety of emergent clinical settings. In addition to 2 method of use patents, we are pursuing new composition of matter IP for the intravenous injectate. On the right, preliminary experiments in mice show it's not only feasible, but it's as effective as a direct myocardial injection, even when it's administered 20 minutes after the initiation of reperfusion intravenously. This is highly clinically relevant. Again, this is 1 dose at 1 time. We also know it's stable at room temperature in lyophilized form and readily soluble in saline, so it should be suitable for off-the-shelf storage and use. And so we anticipate that that part of the project should proceed pretty quickly. We are working up the mouse data, the IV construct data, as well as the rabbit data, and we'll give this to Anita O'Connor. She has decades of experience in regulatory guidelines, FDA regulatory guidelines. So she's going to advise us on the specifics so that we can assure compliance and also future endpoint acquisition requirements so that we can position ourselves to be ready for a pre-IND meeting with the FDA. My goal is to complete these preclinical studies and then work with a CRO to validate that's in a larger preclinical animal model so that we can then hand it off to larger operation to manage the human clinical trials. So at this point, we are working up the rabbit data to present to the NCBC. We are working to refine the formulation to generate the viable IV candidate. And we are working with Tasha Garcia and hopefully the Children's Heart Foundation. I should find out soon. We are exploring to explore the unexpected congenital heart findings further. If things proceed as they have, and it looks like FRNA1 is potentially actually an orphan drug for this rare pediatric disease, we'll consider applying for the FDA voucher program. Every 40 seconds in the time someone has a heart attack, and the time it's taken me to present this work to you, 15 more people have suffered an MI. Vital Effects 1 achieves excellent results. This is 1 dose at 1 time. We're developing this technology to alleviate the socioeconomic impact of cardiovascular disease, and we aspire to make this first of its kind treatment available to patients so that we can preserve their quality of life and longevity. We still have a lot to do, and I estimate that it will take on the order of 3.75 million just to solidify the translational case before we go to larger animals. Our objective in participating in this program is networking, hoping to garner interest and involvement of those with the passion and the expertise to scientifically, commercially, and hopefully also financially support this endeavor. We need to build out our team of advisors and consultants and form partnerships with those who can help us lead the vital effects charge. And hopefully together, we can change the course of cardiovascular disease outcomes. Join me in honoring those who've suffered and give hope for the future to those who are at risk now. I'd really like to express my sincere gratitude to my personal and professional spheres of influence. Some of them I've shown you here today. The support that I've gotten from the ECU Research Enterprise, and all of you for being here today, working with us to equalize the playing field for academic women entrepreneurship. Thank you.